Hello, welcome to the course, Science Fair 101. This is Module 2 of the course, and this section discusses some examples of chemistry science fair project ideas for middle school students and high school students. If you enjoyed watching this video, click on like, subscribe, the bell, and share below so that you can receive notification of new videos I post in this channel. Here are a list of 7 example science fair projects that are suitable for middle school students that I discuss in this video. The first project involves using red cabbage juice as a pH indicator, and the second project involves measuring the time of fizzing of antacid tablets. The third project involves growing salt and sugar crystals and their formation at different temperatures, and the fourth project involves determining what kind of coatings can be used to inhibit the formation of rust in steel nails. The fifth project involves redetermining the effectiveness of different homemade antibacterial solutions on the inhibition of bacterial growth. The sixth project involves the formation of biodegradable plastic from milk. The last project involves looking at the surface tension with and without soap. In this section of the video, I will present the 7 chemistry science fair projects I mentioned earlier, ideas that are especially suitable for middle school students. On the right side is a picture of red cabbage that has been cut open. This project involves first blending the red cabbage and then obtaining the liquid extract. This liquid extract is then added to a variety of household liquids such as baking soda solution, vinegar, soap, soda, and other liquids or solutions. There will be color changes that will be observed. Now, take a photograph of the solutions showing their colors. Then map the colors based on a pH color chart. Basically, the experiment shows that red cabbage extract can be a useful natural pH indicator. This is a qualitative experiment, so the data obtained is simply the colors of the solution that is photographed versus a picture of a pH color chart. The colors can be matched up, and it can be determined if the pH measured using the red cabbage pH indicator accurately reflects the actual pH of the liquid. This can be done by comparing the photographs to a litmus paper dipped in each of the solutions. The picture on the right shows antacid tablets. In this experiment, the goal is to determine the amount of time for the fizzing to stop in simulated gastric juice, which is basically diluted hydrochloric acid. Here, you can experiment with different brands of antacids as well as the form of the antacids such as tablets, chewable, and powder form. There will be a noticeable difference in the amount of time that the gastric acid solution fizzes for these different cases. In this experiment, the fizzing analysis can be conducted 3 times for each sample, and the mean and standard deviation of the time for the fizzing to stop can be determined and reported in the analysis of the results. This experiment involves growing macroscopic salt and sugar crystals. It involves first preparing supersaturated solutions of salt or sugar and placing them in a jar at room temperature, heated in a hot plate, or chilled in the refrigerator. A string is suspended in each of the 3 jars containing the supersaturated solutions. After 3 to 5 days, observe how the crystals look in each case and draw conclusions on how heating, cooling, and normal room temperature plays a role on the crystal formation. There will be clear difference in the 3 cases. In this experiment, the amount of salt or sugar crystals formed can be measured and compared for the 3 cases. The experiment can be done with 3 sets of samples for each temperature condition. The size of the crystals that are formed can be measured, and the average and standard deviation of the crystal size can be obtained for salt and sugar crystals formed at room temperature, when heated in a hot plate, and when chilled in the refrigerator. There will be obvious results, and these results can be analyzed to draw conclusions. Point 0. This experiment involves the use of a variety of materials such as oil, paint, vinegar soak, and salt spray as coatings on steel nails. The study is to determine which coatings are best to use to prevent rust formation. This experiment is more qualitative, and the results can be pictures of the steel nails with the different coatings initially and after a few days. The pictures would clearly show which coating or coatings are the most effective to prevent rust formation. This is a hands-on interactive experiment to see the effect of cleaning the surface with antibacterial soap, vinegar, or rubbing alcohol, and then swabbing the agar plate results in colony formation. A control solution would be an unclean surface swabbed onto agar. The goal in this experiment is to let the bacteria grow in the agar plate and then count the number of colonies formed in each of the different conditions. The average number of colonies and the standard deviation can be determined for each of the different cases and compared to the unclean surface, which should have the highest amount of colonies present. This experiment involves the production of casein pistic, which is a biodegradable plastic which has different tensile strengths depending on the milk-vinegar ratio. The tensile strength can be measured at different milk-vinegar ratios. The average tensile strength and standard deviation can be calculated at various ratios of milk-vinegar. This experiment involves looking at the surface tension of water drops without soap versus water drops with different brands of soap. The goal is to see which ones are able to float a paperclip on top of the bubble. You would record the results on whether the paperclip floats in each case or not. This experiment is a more qualitative experiment to understand the concept of surface tension. Basically, surface tension is the tendency for a liquid solution to maintain its surface area. In this section of the video, I will focus on the different types of experiments that can be done as science fair projects for high school students. One example involves the preparation of hydrogels and monitoring their swelling based on the pH of the buffer solution. Another project involves measuring the enzyme kinetics in which catalase is used as the enzyme and the enzyme activity is measured at different pHs. Yet another experiment involves the creation of fruit cells and compares the performance of these batteries to other types of batteries. Another project involves monitoring the adsorption of heavy metals by activated carbon. Also, biodiesel production from vegetable oil is conducted using a variety of different catalysts. Another interesting project involves determining the caffeine concentration in several popular beverages. Last but not least, gold nanoparticles are synthesized using the sodium borohydride reduction method, and the optical properties are measured. A common method of synthesizing hydrogels involves using acrylic acid monomer, MBA crosslinker, and APS as the initiator. This results in the formation of hydrogels. The hydrogels are soaked into buffer solutions of different pHs such as 5, 7, 9, and 11, and the effect of the pH on the swelling ratios of the hydrogels are determined. One common application of hydrogels is in contact lenses as shown in the right, where a woman puts on contacts in her ears. First, a calibration curve is plotted for caffeine standards of different concentrations using a spectrophotometer and monitoring the UV absorbance at 273 nanometers. The absorbance versus concentration plot is conducted for determining the concentration of caffeine in unknown samples. This results in a linear calibration curve. The coffee, tea, and different soda samples are analyzed using the spectrophotometer at the 273 nanometers wavelength. If the absorbance is higher than the calibration curve limit, the samples should be diluted, and then after the concentration has been determined from the calibration curve, the concentration is multiplied by the factor of dilution. On the right is a picture of a cup of coffee and coffee grounds next to it. This experiment involves determining the catalase enzyme activity at different pH. A potato extract is used as the catalase enzyme source. It is added to different buffer solutions, and then 3% hydrogen peroxide is added to the solution. Then a water displacement apparatus or a gas syringe is used to collect the oxygen volume over a fixed time. The kinetics of the reaction can be followed at different pH, and a calibration curve can be plotted. The goal of this project is to determine which fruit or vegetable, lemon, potato, or apple, results in the highest voltage and current in the ZNCU galvanic cell. The experiment involves inserting the electrodes into each fruit and measuring the voltage and current and the power output. The average voltage, current, and power output, as well as the standard deviation, can be calculated. Based on the results, you could determine which fruit is the best to use for ZNCU galvanic cells to generate the highest voltage, current, and power output. This project can be done if you have access to an atomic absorption spectrometer at a university nearby. In this project, the adsorption of Pb+2 onto activated carbon is assessed at different concentrations of lead-2 nitrate. The Pb+2 source is allowed to mix with the activated carbon for a few hours, and then the solution is filtered out. The remaining solution contains unadsorbed Pb+2 ions, and the concentration can be determined by using atomic absorption spectroscopy, in which a calibration curve is prepared with Pb+2 standard solutions of different concentrations. The concentration of the remaining Pb plus 2 ions detected by atomic absorption spectroscopy in the filtered solution can be subtracted from the total Pb plus 2 concentration to determine the amount of Pb plus 2 that is adsorbed onto the activated carbon. The average Pb plus 2 concentration and standard deviation that are adsorbed onto the activated carbon support material can be reported. This is a project involving obtaining high biodiesel yield using the transesterification reaction catalyzed with NaOH, KOH, or CHO with canola oil. The question is, which of the 3 catalysts results in the greatest yield of the biodiesels? The biodiesel layer is separated, washed, dried, and weighed. The average and standard deviation of the yields can be calculated, and the purity of the biodiesels can be assessed by doing a titration experiment. This experiment involves the synthesis of gold nanoparticles using the sodium borohydride reduction method. Sodium borohydride is the reducing agent, cetyl trimethylammonium bromide, CTAB, is the capping agent, and chloroauric acid is the precursor metal source. The figure on the right shows an artistic rendition of gold nanoparticles that are conjugated with molecules. If access to a UV-visible spectrometer is available, the absorption spectra can be measured and the surface plasmon band can be observed. If a transmission electron microscope, TEM, is available, images of the 2 to 3 nanometers gold nanoparticles can be observed. This project is more of a qualitative project and involves the synthesis of the gold nanoparticles and characterizing them. These are the results that you would present in this project. Overall, the slides in this video detail 7 examples of chemistry science fair projects for middle school students and 7 examples of science fair projects for high school students. If you enjoyed watching this video, click on like, subscribe, the bell, and share buttons below to receive notifications of future videos that I post on the channel.